Hello, I am Pushpangna from IIT Kanpur. Today I'll be discussing the exciting results of my PMRF project on elucidating the entry and fusion landscape of coronaviruses. My PMRF project has been to delineate the conformational dynamics of the spike glycoprotein trimer during fusion and entry. The spike glycoprotein trimer is the major determinant of host tropism. It has a receptor binding domain and the fusion domain. The receptor binding domain interacts with the host cell receptor, and then the fusion domain changes its conformation so that it can latch onto the host cell membrane, making the interaction a stable one. And once that happens, the two membranes are fused together and the viral genomic material is delivered inside the host cell. So, the first research question for my doctoral thesis was to find out the triggers for entry and fusion of coronaviruses. Now, the story has already been completed, and I showed the data in my first and second reviews. The major findings from this project are: first, low pH and calcium trigger spike conformational change for membrane fusion, and second, evolved spike variants show correlation in calcium sensitivity and fusion activity. This work has already been published in the journal Cell Reports. This brings me to my second exciting story, which is to find out the conformational changes of the spike when it is interacting with a phospholipid during membrane fusion. This work is divided into two parts, and I have already shown the data of the first part in my previous review. The key findings from the first part of the review are: first, lipid mixing for SARS-CoV-2 membrane fusion requires phosphatidylserine or PS, and second, SARS-CoV-2 fusion to host cell is phosphatidylserine or PS dependent. Having established the role of phosphatidylserine in fusion, I next decided to jump on to the next question in the project, which was to find out if phosphatidylserine can directly interact with the spike. For that, I developed a single molecule FRET assay or an SM-FRET assay that could report on spike-PS interaction as a function of change of FRET states. A pegylated quartz slice was used to immobilize virions using biotin streptavidin interaction. The pseudovirions were prepared using orthogonal translation system, which had size 5 at the 836 position of the spike protein. This 836 position is the FPPR region or the fusion peptide proximal region. SM-FRET imaging to monitor direct interactions of PS with the spike was carried out using liposomes with NBD-PS. So here NBD-PS will act as a donor and the Psi5 in the spike glycoprotein will act as the acceptor. So the NBD-PS concentration in liposomes was maintained at 0.6 nanomolar in order to ensure that only a single NBD-PS molecule interacts with the spike. So if PS interacts with spike, it will result in a high FRET state. However, if PS does not interact with the spike, it will result in a low FRET state. So when I perform the SM-FRET imaging in a non-equilibrium state in presence of calcium and low pH, interestingly I observed that SARS-CoV-2 spike transitions from a low FRET state to a high FRET state by interacting with PS. Now, as can be witnessed from the data here, which is for the delta spike as well as the D614G spike, I observed a direct change in FRET state from zero point 0.2 low FRET state to zero point 0.9 high FRET state with many intermediate FRET states in between. So this change in FRET state is a result of interaction of the spike with phosphatidylserine. So this is a representative trace of the non-steady state imaging that I performed. Here the acceptor is in red and the donor is in green. So from here, I was able to conclude with very solid experimental evidence that SARS-CoV-2 spike and the spike glycoprotein only directly interacts with phosphatidylserine through its fusion peptide. I next sought to understand that if calcium is removed from the system, does the spike revert to its pre-fusion conformation? So I designed a different SM-FRET assay in order to understand whether the post-fusion conformational change of the spike is reversible or irreversible. So the same pseudovirions and proteoliposomes were used. However, once the virus liposome complex was incubated at low pH and calcium, after that, all the calcium from the system was chelated using EDTA. So EDTA incubation was done for 15 minutes and then imaging was done, and later it was done for 30 minutes and then imaging was done. So as can be seen from the contour plots here, this is the contour plot for imaging at pH 4.6 and calcium. Then after EDTA incubation for 15 minutes, then after EDTA incubation for 30 minutes, So as can be seen from all the data, the high Fred post-fusion conformation did not revert to any other Fred state, showing that the post-fusion conformation is thermodynamically stable and irreversible. So this is one of the representative traces which can be seen. So hence, in the presence of a target membrane, for example, such as the membrane of the host cell or membrane of the liposome in my assay, the post-fusion conformation that is formed is highly stable and irreversible. And that is what which leads to fusion of the virion with the host cell. So the next step was to understand if virus internalization is dependent on PS or not. So for which I designed a cell-based virus internalization assay. So here Vero TMPRSS2 cells were used as a target cell line and they were transfected with LAC-C2 which was used to MRFP. So LAC-C2 expressed inside the cell binds to PS on the inner leaflet of the plasma membrane and prevents PS flipping. Hence the cells in which LAC-C2 is being expressed, that is the cells which are red, which can be seen here and here. So technically they should not be able to internalize viruses. So this can be seen from the data here. Here the white arrows are showing virions inside cells which are not red, that is where LAC-C2 is not being expressed. So here wherever LAC-C2 is present, there we do not see any internalized virions. As a control for the internalization assay, which showed that PS virus internalization is dependent on PS, so I performed this antibody staining to ensure that AC2 and TMPRSS2 that is being expressed in target cell is not affected by LAC-C2 expression. And the result is here. So this is the antibody staining for AC2 in absence of LAC-C2 expression and in presence of LAC-C2 expression. And this is the data for TMPRSS2 in absence and presence of LAC-C2 expression. So from all both the data, it can be seen clearly that LAC-C2 expression does not affect AC2 or TMPRSS2 expression in target cells. Now, in order to control for the non-specific negative charge requirement of SARS-CoV-2 fusion, because as we all know, PS is a negatively charged lipid, I substituted the PS and liposomes with another anionic phospholipid, that is PG or phosphatidylglycerol. glycerol. So I performed the bulk fusion assay where pseudovirions were labeled with the lipophilic dye DIO, and this fusion reaction was observed as a rate of dequenching of di-DIO when it is fusing with the liposomes. And I found that PG had somewhat similar effect as PS. However, the effect with PG is reduced as compared to that of PS. Now, if we compare the fusion efficiency for D614G virions and the delta virions, in D614G, when we are using the PG lipid for fusion, the fusion efficiency is reduced to be somewhere around 4 to 5% as compared to the efficiency that is around 30 to 37% in presence of PS. In the delta strain, however, the fusion efficiency in presence of PG is reduced to around 40% as compared to the 90 to 95% efficiency observed in presence of PS. This no PS or no PG control has been used to show that the anionic phospholipids are playing a very important and crucial and significant role in mediating fusion. However, here it is very important to mention that only 1 to 2% of PG is present in mammalian cell membrane, whereas PS is present in abundance in mammalian cell membrane. This brings me to the model that I have proposed based on all the data that I discussed right now. So according to this model, the viral spike interacts with AC2 and TMPRSS2 on the host cell membrane, and the virus is then endocytosed. And during late endosomal maturation, when the pH is low, its conformation changes and the fusion peptide interacts with phosphatidylserine, following which membrane fusion takes place. And this is the first time that the research work has shown directly that PS can interact with the viral spike, and interaction of PS with the spike is crucial for fusion to take place. This work has already reached its first review stage in Nature Communications. So these are the three major conclusions from my work. The first is that low pH and calcium trigger spike conformation change. This work has already been published in Cell Reports, and these two conclusions, like the paper for it is in first review stage in Nature Communications, and here I have established that phosphatidylserine interaction is crucial for fusion for coronavirus membrane fusion to happen, and that a target membrane is essential for stabilization of the post-fusion state of the coronavirus spike. These are the publications which have been resulted from my work, and this year I presented my work in a national conference in AITH, and I received the first prize for my work, or the Research Excellence Award. And also, I went to GRC, the Physical Virology GRC, in January, and I was invited for a student talk there. And I also presented a poster on the basis of my student talk. I was awarded the Carl Storm International Diversity Award. That is all. Thank you. I'll be glad to take any questions.